So good afternoon, and thank you very much for this kind invitation to this so important symposium on psoriasis. So it's a pleasure to share our work on IL-34 expression and function on HS and psoriasis. So IL-34 is a cytokine produced by keratinocytes with important role in epidermis. Indeed, it helps to maintain Langerhans cells in the skin. In psoriasis, the regulation of this cytokine has been reported with also a loss of the Langerhans cells. So far, there are no existing data in HS. Therefore, the aim of this study was to investigate IL-34 expression, regulation, and function in HS and psoriasis. To achieve this, we used lesional skin and non-lesional skin from psoriasis in HS. We performed a bulk RNA-seq, single-cell RNA-seq, plus integrating this data with immunohistochemistry, while for function validation in vitro, used human keratinocyte, generating 2D and 3D models. We found that IL-34 is down-regulated in lesional skin respect to non-lesional skin by bulk RNA-seq, but from single-cell analysis, we also noticed that in no psoriatic skin, the major source of keratinocytes are a subpopulation of keratinocytes. In particular, the differentiated keratinocytes are the major source of IL-34, with a marked reduction in psoriasis and also in HS. As you can see, in healthy was mainly distributed in upper layers. So then for the function, we isolated T cell from psoriasis skin and we cultured. These supernata then were used to co-culture 3D model to see the effect of Th17 environment, and as a comparison, we also used IL-22, a canonic anti-differentiative cytokines. As you can see by RNA-seq data, IL-34 is mainly down-regulated by Th17 environment. So then we also used a 2D model to understand also if the production of this cytokine is increasing during the keratinocyte differentiation. Therefore, we used our protocol using a terminal differentiated keratinocytes model, where we noticed that 4 days post-confluency, there is a huge increase of this cytokine. Therefore, at day 4, we also stimulated with the emerging cytokine in HS, for example, IL-17A plus F, combining also with IL-22. As you can see, there is a strong downregulation, so these cytokines are the main involved in the regulation of IL-34, and with a strong downregulation if they are combined all together. So for the function, so we aimed then, since also we noticed that its main receptor is increasing during this differentiation, we stimulated the cells with a recombinant, a human recombinant, IL-34. As you can see, this cytokine is able to activate the STAT3 pathway, and so provide a new function, so that IL-34 is able also to activate not only, regulate the skin immunity by modulating the environment and also the homeostasis of Langerhans cells, but also be able to activate the keratinocytes in autocrine and paracrine manner. In particular, we have used also a pathway enrichment analysis, and we noticed that below this signaling, so after the activation of STAT3, can be involved a lot of the tight junction and also proteins that are important for the skin formation. In particular, now we found also a main target to IL-34 that is a part of the Claudin family. So IL-34 can be restored, so it can be a potential negative marker of then related to the progression of the disease that can appear after 8 weeks of secukinumab therapy, but can be also be able, thanks to its property, to restore the repopulated Langerhans cells in the epidermis. This was also possible in patients that they can reach 80% of the reduction in PASI score respect to the baseline. So in summary and conclusion, IL-34 and keratinocyte differentiation marker are reduced in HS and psoriasis lesions compared to non-lesional skin. IL-17A/F, IL-22 impair keratinocyte differentiation, thereby also IL-34 production. IL-34 activates STAT3 in differentiated keratinocyte, potentially supporting skin barrier. Therefore, restoration in responsive patient after Th70 blockade suggests are establishing skin homeostasis and also barrier integrity. I want also say thanks again to SDR and the PC Symposium for the invitation, also the collaborators, in particular Kilian Eirik, and my mentor, Cristina Baresi, who is constantly supporting me. Thank you.
Any questions for Dr. Scala? No?
Was there any differential expression in the different sides of HS lesions? Because, you know, it's quite heterogeneous.
It's quite heterogeneous, and one of the critical points also where to take biopsies for these patients, right? So at the moment we're using a bulk RNA-seq from a public available data, but we want to also generate our data set and also to define some criteria, because at the moment it's mostly significantly downregulated in psoriasis, that is, that there is this hyperproliferation of keratinocytes. In HS, it's depending. It's not so downregulated, but still, it is. So it could be also interesting because there are no data regarding IL-34 in HS.
Thank you.
Okay. Thank you so much. Thank you.
you.
And good luck with your further research.
Thank you.